Today we'll talk about the mechanism of transcription. And it's mechanism of transcription. This transcription, we've already discussed what the transcription is. Regarding the mechanism of transcription, it is a multi-step process. Several steps are involved in this process. First step is activation of ribonucleotides. The second is activation of ribonucleotides. The second step is binding of RNA polymerase to DNA duplex. The third step is exposure of DNA bases. Then next step is complementary base pairing. Then next is the formation of RNA chain, and finally we have termination of RNA synthesis and release of RNA chain. These are some of the important steps which are involved in the synthesis of RNA chain, that is transcription. So we'll discuss these steps one by one. As you know that transcription is simply the synthesis of RNA from the DNA. Regarding the first step, activation of ribonucleotides. Activation of ribonucleotides. These ribonucleotides, these are basically the building blocks of the RNA. So ribonucleotides, they are the building blocks of RNA. Four types of ribonucleotides, they participate in RNA synthesis. One is adenosine triphosphate, guanosine triphosphate, cytidine triphosphate, and uridine triphosphate. So we can say that we have got four types of ribonucleotides, and these ribonucleotides, they have to get incorporated so that they will get involved in the RNA synthesis. For that purpose, their activation is required. And how they are activated? They are activated through phosphorylation reaction, where an enzyme phosphorylase and energy from the ATP is utilized for the conversion of ribonucleotide monophosphates to ribonucleotide triphosphates. This step is called as activation of ribonucleotides. So we have got these ribonucleotides. They are building blocks of RNA. These are building blocks of RNA. So four types of ribonucleotides participate in RNA synthesis. We have adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and uridine monophosphate. You know that in case of RNA we have got uracil, while as in case of DNA we have got thymine plus H3PO4 phosphoric acid. H3PO4 phosphoric acid, and then there is an enzyme that is called as phosphorylase. Phosphorylase, and energy from ATP. It is utilized for the activation of these ribonucleotides. So we will get adenosine triphosphate, guanosine triphosphate, cytidine triphosphate, uridine triphosphate, and this reaction is called as phosphorylation reaction. And this H3PO4, this is phosphoric acid, H3PO4 phosphoric acid. So these ribonucleotides have to get involved in the synthesis of RNA, therefore they are activated. They are converted from monophosphate to triphosphate so that they become the building blocks of they become the substrate for the RNA synthesis. Then we have got second point is binding of RNA polymerase to DNA duplex. Binding of RNA polymerase. We have first Then second one is, this was first one, binding of RNA polymerase to DNA duplex. Binding of RNA polymerase to DNA duplex. We know that the main enzyme for the RNA synthesis is RNA polymerase. As we have got in case of DNA, the main enzyme for DNA synthesis is DNA polymerase. Same is the case we have got in case of RNA synthesis, the main enzyme which is involved in RNA synthesis, that is RNA polymerase. So this RNA polymerase, it gets attached to DNA duplex. Actually, we have got a promoter region. Promoter. This promoter region is present on the DNA duplex, and this promoter it acts as a binding site for the RNA polymerase. So promoter, it is a binding site for RNA polymerase. This promoter of the DNA duplex, it is a binding site for RNA polymerase. We have a factor. Actually, this RNA polymerase, it has several subunits. It has alpha, alpha dash, omega, and sigma. So this sigma factor, this RNA polymerase, so many subunits, and one of the subunit of the RNA polymerase is sigma subunit. This sigma subunit helps in recognizing this promoter of this DNA duplex, and also helps in the binding. That is, it helps in the binding of RNA polymerase to the promoter of the DNA duplex. So sigma subunit. This sigma subunit of RNA polymerase helps in binding of RNA polymerase to promoter. This sigma subunit, it basically recognizes this promoter sequence and also helps in binding of RNA polymerase to DNA duplex. Then we have got third step is, we have exposure of DNA bases. Exposure of DNA bases. This exposure of DNA bases, we know that this DNA, it is double-stranded molecule. It is like this. It is a double-stranded molecule. This is a DNA duplex. This is DNA duplex, and whereas RNA is single-stranded. This is single-stranded RNA. Single-stranded RNA. So it is necessary that the two strands of the DNA, they will get separated, they will unwind, they will unzip, so that one of the two strands that will act as a template for the synthesis of the RNA. As RNA is single-stranded and DNA is double-stranded, so only one strand of the DNA, what is called as template strand, that will act as a model, that will act as a template for the synthesis of the RNA. So it is the RNA polymerase. This RNA polymerase helps us in the unwinding of DNA bases. Unwinding of DNA bases. It helps in the unwinding of, unzipping of DNA duplex, we can say. So then we have got why it is necessary for the separation of the two strands of the DNA, because RNA is single-stranded. It means that only one strand of the DNA will act as a template for the synthesis of the RNA, because RNA is single-stranded. Then we have got another point. So then base pairing. We have discussed 3 points. Then fourth point is then we have got next one, that is base pairing. This is fourth one, base pairing, complementary base pairing we can say. Complementary base pairing. This complementary, what do you mean by complementary base pairing? This complementary base pairing is according to Watson and Crick base pairing rule. That means adenine pairs, in case of DNA, adenine pairs with uracil, uracil pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine. This is complementary base pairing rule. So it means that what is important, that it is according to Watson and Crick base pairing rule. Adenine pairs with uracil, uracil pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine. So there are 2 hydrogen bonds between adenine uracil and 3 hydrogen bonds between guanine and cytosine. It means that this RNA polymerase, this RNA polymerase, this RNA polymerase, it breaks the hydrogen bond. So we have got this is double stranded DNA. This is double stranded DNA duplex. So these are hydrogen bonds between the 2 strands of the DNA. These are hydrogen bonds. What are these? Hydrogen bonds. The 2 strands of DNA. This is one strand of DNA, 5' to 3'. This is 3' to 5'. This is one strand of DNA. This is another strand of DNA. These 2 strands of DNA are joined by means of bond that is called as hydrogen bond. This RNA polymerase, it breaks the hydrogen bond between the 2 strands of the DNA, between the 2 strands of the DNA, and then helps in polymerization. They'll help us in linking of these ribonucleotides to form the RNA chain. So in addition to this, these ribonucleotide triphosphates, when they are joined, their pyrophosphate, these ribonucleotide triphosphates, they release their pyrophosphate group. So we can say that ribonucleotide triphosphates, they become ribonucleotide monophosphates. These ribonucleotide triphosphates, they become ribonucleotide monophosphates. For example, we have adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, uridine monophosphate, because these ribonucleotide monophosphates are the actual substrates for the RNA. They are the actual building blocks of the RNA. So actually this ribonucleotide triphosphate, it releases the pyrophosphate group. It releases what? Pyrophosphate group. So what I mean to say is that this RNA polymerase, it breaks the hydrogen bond between the 2 strands of the DNA, and it also helps in linking of these ribonucleotide triphosphates to the DNA bases of the template strand, and ultimately these ribonucleotide triphosphates then release their pyrophosphate group and get converted into the ribonucleotide monophosphates. These ribonucleotide monophosphates are the actual building blocks of the RNA. Now the question arises that why these ribonucleotide triphosphates, then why these ribonucleotides are converted into ribonucleotide triphosphates? Because of the fact that these ribonucleotide triphosphates, in addition to acting as a substrate, these ribonucleotide triphosphates, in addition to acting as a substrate for RNA synthesis, they provide energy. They provide energy in the form of pyrophosphate group for linking, for polymerization of adjacent ribonucleotide to form the RNA chain. So we can say that for example, we have DNA. This will, for example, this is a template DNA. Now ribonucleotide will get attached to here. They will get attached to here. The ribonucleotides. These are ribonucleotides. They will attach to the template DNA according to the Watson-Crick base pairing rule. This is the template DNA, and they will attach to the ribonucleotides according to the Watson-Crick base pairing rule. This will be like this. It can be explained. We can explain like this. For example, we have got a double-stranded DNA. We have got double-stranded DNA. We've got double-stranded DNA. This is DNA duplex. This is, for example, 5' end, 3' end. This is 3' end, 5' end. Suppose this is the promoter. This is promoter. Suppose this is promoter. This promoter will act as a site where RNA polymerase will bind. Suppose this is terminator, transcription, termination site. Transcription termination site. This transcription termination site. These are two strands of DNA have re-annealed. This is complementary base pairing. So these are nitrogenous bases. They are complementary base pairs on the another strand. So for example, this is called as template. This which has polarity 3' to 5'. That will act as a template strand. Which strand will transcription always proceeds in 5' to 3' direction. That means the direction of transcription. The strand which has polarity 3' to 5', that will act as a template strand. That means strand with 3' to 5' direction will act as, that will be a template strand. Template strand. This template strand is also called as antisense strand. Template strand is also called as antisense strand or non-coding strand, as we have already discussed it. Template strand is also called as antisense strand, whereas non-template strand is called as sense strand or coding strand, because it is the sequence of non-template strand is similar or identical to the mRNA chain. Therefore, we say that this non-template strand is called as coding strand. So these are two DNA strands, two strands of DNA have re-annealed. rejoiner. Two strands of DNA have annealed, and this is newly synthesized. Suppose this is newly synthesized RNA. So we can say that newly synthesized RNA on DNA template. Newly synthesized RNA on DNA template. This is newly synthesized RNA on DNA template. So this is base pairing between DNA and RNA, DNA and RNA. And this base pairing is according to Watson-Crick rule. Adenine pairs with uracil, uracil pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine. So suppose this is the direction of transcription. That means direction of transcription. Transcription always proceeds in 5' to 3' direction. So the strand which has polarity 3' to 5', that will act as a template strand. Direction of transcription. Then we have these are, suppose these are, well, this is G, C, U, A, C. These are ribonucleotides. Ribonucleotides. These ribonucleotides are the building blocks of the. These ribonucleotides are the building blocks of the RNA. So this is template strand. This is what template strand. This is obviously, this is non-template strand. Non-template strand. This is template strand and this is non-template strand. So this, we can say that it is a mechanism of transcription. So we were discussing about these ribonucleotides, ribonucleotide triphosphates. These ribonucleotide triphosphates, they join to the, these ribonucleotide triphosphates, they join to the DNA bases according to Watson-Crick base pairing rule. After that, these ribonucleotide triphosphates, they release their pyrophosphate group and they get converted into ribonucleotide monophosphates. The energy which is released during this process, it is utilized by the RNA polymerase for the polymerization, for the linking of free ribonucleotide monophosphates to form the continuous RNA chain. And this RNA chain is present on a DNA template. And these ribonucleotide monophosphates, they are actually the actual building blocks of the RNA. Then we have got another important point, that is formation of RNA chain. This was 5th point, formation of RNA chain. This is formation of RNA chain. Formation of RNA chain. We know that this RNA polymerase, it links the free ribonucleotides, ribonucleotide monophosphates, and ultimately forms the continuous RNA chain. This is what is called as formation of RNA chain. That means these free adjacent ribonucleotides, these free adjacent ribonucleotides are this RNA polymerase, with the help of magnesium, with the help of magnesium, with the help of magnesium or in the presence of magnesium, it forms a phosphodiester bond between the adjacent ribonucleotide monophosphates, so that this RNA chain will become continuous. And this RNA polymerase, it will form phosphodiester bond between it forms phosphodiester bond between adjacent ribonucleotides. And this RNA polymerase, it forms a phosphodiester bond between the adjacent ribonucleotide monophosphates, so that this RNA chain becomes continuous. And this RNA polymerase, it requires magnesium for this process, for the formation of continuous RNA chain, so that this RNA chain will become continuous. Then we have got another important point, that is this is sixth, termination of RNA synthesis and release of RNA chain. Termination of RNA synthesis and release of RNA chain. This RNA polymerase, when it reaches to the transcription termination site, when this RNA polymerase, so we have got this is RNA polymerase. This is RNA polymerase. This represents the RNA polymerase. This is RNA polymerase. This RNA polymerase, the main enzyme for the RNA synthesis, when this RNA polymerase reaches the transcription termination site, then transcription stops. Actually, this transcription termination site, it is transcription. When RNA polymerase reaches the transcription termination site on the DNA duplex, transcription termination site. This transcription termination site, it is present on the DNA duplex, and when this RNA polymerase reaches the transcription termination site, the transcription stops. This transcription termination site, it has a special sequence which is guanine cytosine rich. It has guanine cytosine rich base pair. That means this sequence, it is rich in guanine cytosine base pair. So it is guanine cytosine rich. This transcription termination site, it is rich in guanine cytosine base pair. When this RNA polymerase reaches this transcription termination site, this transcription stops. There is an important factor that is called as Rho factor. Rho factor. This Rho factor helps in the termination of transcription. It helps in the termination of transcription. This Rho factor, it is Rho factor. This Rho factor, it binds to this Rho factor. It binds to the RNA chain. When it binds to the RNA chain, this Rho factor, it acts as an ATPase and stops transcription. This Rho factor is also responsible for the dissociation of this RNA chain from the DNA template. Now, so we can say that this when this RNA polymerase reaches the transcription termination site, this transcription halts. It stops. There's a factor that is called as Rho factor. The Rho factor plays an important role in the termination of the transcription. This Rho factor, it binds to the RNA. This Rho factor, it binds to the newly synthesized RNA chain at the transcription termination site. And this Rho factor, it acts as an ATPase and stops the transcription. This Rho factor, it is also responsible for the dissociation of the RNA chain from the DNA template. So this indicates that termination of the RNA synthesis. So this was regarding the mechanism of transcription. In the next video, we'll discuss about the post-transcriptional modifications. Thanks.